Hi guys, in this video we're gonna talk about the neuroanatomy of the cerebellum. So the cerebellum is part of your hindbrain that is located in the posterior cranial fossa. When we talk about the cerebellum is actually formed of those kinds of elevations like the gyri, but here we call them folia, and the fissures that is basically between them. We have major fissures that kind of separates the cerebellum into loops. And the most important ones, we're going to talk about them, okay? But no, we're not going to talk about all of them. We roughly have like four fissures in the cerebellum, but the most important ones is the ones that separates your important loops, major loops. The first one is the primary fissure. So the primary fissure is the fissure that divides the cerebellum into anterior loop and posterior loop. And actually, posterior loop is not only this part. If you continue and look from down, the whole thing is also part of the posterior loop. All right, so it's not only this part from the superior surface, it continues all the way to cover the whole inferior surface. So the primary fissure separates your anterior loop, this small part of the superior surface, and the whole posterior loop, which includes this part of the superior surface and the rest of the inferior surface. Now that will take us to the next fissure that separates my superior surface from inferior surface. Now, if you guys look in the superior surface, it's basically the sloping surface. If you hold the cerebellum in your hands, let me see if I can have a better. If you guys look at the cerebellum, you guys can see that this is a sloping kind of surface. This is the superior surface, all right? And the convex, all right, the convex parts here, this is the down part. This is the inferior surface. And that way, you are holding the cerebellum in the correct anatomical position, all right? Now let's get back to the fissures, all right? So the superior view, and this is the convex, the bulging parts that sits in the posterior cranial fossa is your inferior surfaces, and they are separated by the horizontal fissure, all right? So if you look from here, the horizontal fissure here separates your superior surface above from the convex inferior surface below. Okay, so these are two major fissures. Now, what about the third one, which is an important one? It's basically what we call the posterolateral fissure. If you guys trace this area here, there is a fissure that is posteriorly, and laterally. And the posterolateral fissure separates your flocculonodular loop, and we're going to talk about that as we proceed from the loops, all right? So in order to have distinctive flocculonodular loop, we have the posterolateral fissure here. We also have two here fissures, but I don't want to talk about them because that is not going to give me a major part of the cerebellum, okay? So that was general talking about the loops and fissures, and now going into the parts. Now, if you look at the cerebellum, guys, it has two cerebellar hemispheres. Hemispheres means a half of a ball or a sphere. So this is a hemisphere, and this is a hemisphere, and in the center you have something called vermis. All right, so the vermis is in the middle part of your cerebellum. You have superior vermis, which is basically, if you look from the top, this is the superior vermis on the top view in the superior surface, and you have inferior vermis. So if you look at the inferior vermis, it has four parts: the nodulus, the lobula, the pyramid, and tuber. Okay, that's all fine, but the important thing to me is the nodulus, because the nodulus, as a part of the inferior vermis, along with two parts, we call them flocculus, and the flocculus means tuft of wool. It's kind of like a flower kind of thing. And if you guys look at the colored picture, it's basically, they are basically coloring the whole loop, because this is a functional and structural separate loop responsible for balance in the blue color, okay? Because that is basically giving you the nodulus as a part of the inferior vermis, along with those two pieces of flocculi, All right, so together they are flocculonodular loops. Okay, this is the flocculonodular loop. That's why the nodulus is an important part of the inferior vermis. These are the major parts of the cerebellum. You have superior surface, inferior surface. You have two hemispheres. All right, you have the vermis, you have the flocculi, all together with part of the inferior vermis, they will give you the flocculonodular loops. All right, so these are the parts of cerebellum. Now, moving on, there is a very important structure. If we come back to this, if you guys look at the hemispheres, you will find a part that is basically kind of bulging, and it's actually coming out of the contour of the cerebellum. And you guys know that the whole cerebellum is sitting in the posterior cranial fossa, and in the center here down you have your foramen magnum. So these two parts, if there is any increase in the intracranial pressure, if you guys look here, the tonsils can easily bulge into the foramen magnum. Okay, because you know the foramen magnum is the biggest foramen you have in the base of the skull, and you guys know that the brain is sitting in the skull itself. So if I have a neural tissue that is kind of coming out of the contour of the cerebellum, and if I have here increased pressure, that will slip into that opening, which is a bony opening, and that will kind of compress on the important centers in the brain center, like the respiratory center and the centers controlling vital functions in your body. So these tonsils are very important structures that is clinically significant in terms of if there is an increase in intracranial pressure, that will cause tonsillar hernia, and that hernia will cause the tonsils to come out of the foramen magnum, causes life-threatening consequences. And more information about that should be explained in clinical medicine and other disciplines. All right. Moving on, the cerebellum is connected to your brainstem by peduncles. All right, so peduncles are those stalk-like structures that projects out of the cerebellum, and they will give you connections with the cerebellum. So you guys, if you kind of try to envision what happens as in a lateral view, you have here your brainstem, you have your midbrain, the pons, and the medulla, and the cerebellum is actually at the back. All right, so you have superior cerebellar peduncle that connects your cerebellum to the midbrain. You have middle cerebellar peduncle that connects the cerebellum to the pons, and you have your inferior cerebellar peduncle that connects the medulla to the cerebellum. Okay, so we have the superior, it's the medial most one of them, the inferior cerebellar peduncle, which is in the middle portion of it. And then you have the middle, which is the biggest one, and it's kind of hugging and covering them, because this is what connects your pons to the cerebellum, and this is what means pons means a bridge. So it's kind of bridging and kind of covering the other ones. All right, so kind of like that, the blue one, it's the middle cerebellar peduncle. It's basically connecting the pons to the cerebellum, so it's giving you this kind of as if it's hugging the other two ones, and it's kind of covering them from outside. Okay, so this is if we have a section and we remove the cerebellum and we look into the fourth ventricle, because you guys know that the cerebellum is the roof of the fourth ventricle. And remember when we talked about the brainstem and we said that the floor is half part from the pons and the lower half is from the open medulla. That was the floor of the fourth ventricle. Now, what happens if we look at the peduncles, guys? Now, think about your fourth ventricle as a room. Okay, so you have a room here, which is basically your fourth ventricle, because ventricle means a space, and this is one of the spaces the CSF is swimming inside, swimming around the brain and spinal. So the CSF needs to have spaces where they are formed, okay, because we have the ventricles of the brain. These are the widened areas where you have most of amount of the CSF, And in these places, we have the choroid plexus. Choroid plexus is the capillaries, the blood capillaries that will be there in those, the lateral ventricles and the fourth ventricle and the third ventricles, all right, because they are going to form the CSF and they will produce it in that. So fourth ventricle is one of them, which is associated with our topic today, cerebellum, and that is basically a room. The floor of the room is part of it pons and part of it the open medulla oblongata, which we talked about that in the brainstem, and the roof of that is actually a white matter, pieces of neural tissue that is basically filling in the gaps between those cerebellar peduncles. So I have superior medullary velum that connects the superior cerebellar peduncle right and left, and I have inferior medullary velum, which is the pieces of white matter tissue that bridges the gap. So this is what we call the superior medullary velum and the inferior medullary velum. Velum, it's kind of a roof, a kind of tent-like roofs of the fourth ventricle. Okay, so if this is my room, and and I have a covering on the roof of that superior medullary velum and inferior medullary velum, and here is my floor from the pons and medulla. Okay, so this is gonna be my superior and inferior medullary velum. It's basically the neural tissue here that is filling in the gaps between the cerebellar peduncles. So with that, we are grossly covering all the structures that are forming the cerebellum, talking about the loops, anterior and posterior, separated by primary fissure, talking about the surfaces, superior and inferior surfaces separated by horizontal fissure, talking about the two hemispheres and one vermis in the middle, and part of the vermis forming the nodulus, and the nodulus along with the flocculi forming the flocculonodular loop, which is separated structural and functional loop, and we're going to talk about that as we proceed into more details. And then the peduncles, which connect the whole cerebellum into your brain stem by superior, middle, and inferior, and how the superior and medullary velum are forming the roof of your fourth ventricle system. All right, guys, that was just a recap to what we've just mentioned. Now, moving on, we have gray matter and we have white matter. So when we say gray matter, again, we're talking about non-myelinated neurons, or we're talking about collection of cell bodies. And when we talk about white matter, we are talking about myelinated axons, all right, because of the lipids that are forming the myelin sheets around your axons. All right, so white matter is the myelinated axons. You have them running in and out of the cerebellum, because the cerebellum at the end of the day, it wants to do a function, it wants balance, it wants to fine tune your movement, it wants to control your descending motor tracts to perfectly execute motor function. Okay, so there should be inputs and outputs from the cerebellum. We need to kind of ask those guys, how can we form perfect connections, and how can we perform perfect actions? So we need to send them a question, and cerebellum needs to answer our question by sending outputs and controlling how we form the corticospinal tract. So it will give us a feedback to the cortex itself, and it will also give a feedback to the brainstem nuclei that are going to form the extrapyramidal kind of motor descending systems, because at the end of the day, what makes my function is it's gonna be basically, it's whether the pyramidal, which is the corticospinal tracts descending from the big boss, or the extrapyramidal system, and that is going to be another motor system that controls my movement. But that another motor system, it's not from the big boss, it's from the helpers, and those helpers are going to be the cerebellum, the basal nuclei, and the brainstem nuclei. So the brainstem nuclei are actually controlled by the cerebellum and basal nuclei. Okay, so here we have inputs and outputs coming in and out of the cerebellum, all right? and that is basically to control the movement and perfectly execute motor functions. So talking about the organization of the white matter and gray matter, the story in cerebellum is opposite to what we see in the spinal cord. We have the gray matter in the outside. It means the cell bodies and non-myelinated neurons, okay, outside. And we have the axons coming in and out, all right, that is going to be inside. If you guys look at the white matter, which is which is made by the myelinated axons, if you look at it, guys, it looks like a tree, the branching of a tree. So if you trace it, it's going to be a white matter here, and it's going to give you a primary branching, and then secondary branching, and then tertiary, and that gives you an appearance of the arbor vitae. All right, so arbor vitae is basically how in Latin, it's a Latin word for the tree, the life tree. The life tree, this is how the branching of the life tree, and this is how it looks like. So we are talking about the organization of the white and gray matter, and we refer to that as arbor vitae. And this is what does that mean? It means that the white matter, as it gets inside of the cerebellum, it gives you that appearance. Okay, now let's talk about the story of the inputs and outputs in a basic, not molecular, in a basic cellular level. What happens and why those cells and fibers, they look like that? We have axons coming to the cerebellum and axons coming out of the cerebellum. And getting to the cellular level and thinking about the cells, the nerve cells that forms these axons, we have basically three types of neural cells or nerve fibers. All right, these are, two of them are inputs and one of them is the output. All right, now the major output of the cerebellum that is going to give the response of the cerebellum to any information on a question coming to it, it's going to be the Purkinje fiber. And the inputs are going to be either Mosaic fibers or climbing fibers. So what is the difference between the Mosaic fibers and the climbing fibers? There are basically two major differences. One of them is where will they stop? How will they stop those fibers as they come into the cerebellum? The other thing, from where they are coming. Okay, so the Mosaic fibers, let's talk about the stopping of them. The Mosaic fibers, as they get into, let's say that this is a cerebellar hemisphere, and I have inputs coming into the cerebellum. Okay, if the cerebellar input is by Mosaic fibers, it means they are coming from everywhere, and we're going to talk about the everywhere as we proceed. And it's not only that, they stop at the granule layer of the cerebellum. Okay, the Mosaic fibers, they stop at the granule cell layer here. They need to reach the Purkinje. And okay, so let's say that we have molecular layer, and then we have Purkinje cell layer, and we have the granular cell layer, and this is the medulla where you have the axons coming in and out. This is the medulla, which you have the white matter coming in and out. This is the cortex, okay, the gray matter cortex, and this is the white matter medulla. Medulla means axons are coming in and out. Cortex means there will be cell bodies and connections between the fibers. Now, if we talk about the Mosaic fibers, they are coming from everywhere and they will stop at the granular cell layer. They need someone to help them to take the information to the molecular layer. Why do we need to go to the molecular layer? Because you guys, in the molecular layer, you have your Purkinje cell. This is the Purkinje cell that will give you the final output to everywhere. Okay, it will give you the final output. But the dendrites of the Purkinje is in the molecular layer. So in order for the cerebellum to answer you back, you need to give the information all the way to the molecular layer above, because the dendrites of your Purkinje cells are sitting at this level. Okay, so the Purkinje needs to, this will send you the final output and the answer of the cerebellum everywhere. Now I need to ask the cerebellum about the movement, and I have inputs coming from everywhere. The Mosaic fibers, they cannot reach the Purkinje. They are not old enough to reach to them. So they will take a help from the granule cells. The granule cells, once it receives information from the Mosaic fibers, they will ascend and split themselves right and left into parallel fibers. So if you guys look at the granule cells, Mosaic fibers, they will go to them, they will ascend and split themselves right and left. They will try to find any nearby dendritic cells, dendritic fibers of the Purkinje. Why we need the dendrites? Because the dendrites are the receiving parts of the Purkinje. Why do I need Purkinje? Because Purkinje is the final output of the cerebellum. Purkinje fibers are inhibitory. If I'm forming a movement by the cortex, it's gonna be so hyper. Cortex, you guys, is very hyper. It wants to do a movement. It's always hyper. It has positive charge. So in order for the movement to be fine-tuned and perfectly executed, I need to go to the cerebellum and I ask him, please, can you send me some inhibitory signals that makes me doing the movement in more controlled way? So in order for the cerebellum to answer back, you need to reach to the Purkinje cells. So the Mosaic fibers will go ahead and give the information coming from everywhere of our brain centers to the granule cells, and the granule cells will ascend all the way to the molecular layer above, and it will split into parallel fibers. It will try to find any dendritic cells. Once it finds it and gives the information to it, it's done its job. Okay, and that is going to happen with all the brain centers that they want to ask the cerebellum. Okay, so what about the other thing? What is the other thing that you have? The other thing, you guys, I have is the climbing fibers. The climbing fibers are coming from the inferior olivary nucleus. So from the inferior olivary nucleus, we will send input that is called climbing fibers. Why do we call them climbing? Because they have their own stairs all the way to the molecular cells. They have the ability and courage to go all the way and meet the Purkinje dendrites themselves. They don't need anyone to help them. They will go ahead all the way until they reach the molecular layer. They will give the information to the dendrites of the Purkinje, and then the Purkinje will respond to that by sending outputs to them back to the inferior olivary nucleus. All right, so this is the whole idea and story of the inputs and outputs at the cellular level. Let's go ahead and repeat that again. So what happens, you guys? What happens is the Mosaic fibers, they send their information to the granule cell layers, and it will split into parallel fibers, and that is coming from everywhere. So the inferior olivary nucleus, it has its own climbing fibers. This is the other types of input fibers. They have all the ability to go all the way to the dendrites of the Purkinje cells, and the output is going to be by the Purkinje. So Purkinje cells will go all the way, and they will answer you back from wherever you're coming, and they will kind of fine tune your movement, and they will control the information coming to them. All right, so remember that the QT olive, all right, the inferior olivary nucleus, they like to climb the stairs. They don't need helps. They have their own stairs that takes them all the way to the molecular layer, to the because in the molecular layer you have the dendrites of the Purkinje cells, and once you affect them, you will be able to have a response from the cerebellum. All right, guys, so this is what happens, and this is another histological pictures to show you guys why do we call this molecular, because it has all of these dendritic cells, and here Purkinje cell is embedded in the Purkinje layer, and then you have the granule cells, because the cells, they look like granules. Can you guys see? It looks like a granules kind of area. All right, so this is basically the cell bodies of the granule cells. This is where your Mosaic fibers are going to stop to give the information here, and once you get into the granule cells, you will give your the Mosaic fibers. Mosaic fibers, they will give the information to them to help them, and they will go up to give those parallel fibers right and left to find the dendrites of the Purkinje, any nearby Purkinje, and then you will find a response as a final output, and the only output is coming from the Purkinje to everywhere, and the MOSE fibers will be from everywhere except inferior olivar nucleus, and the inferior olivar nucleus and the medulla. They have their own stairs, so that's why we call them climbing. They can climb all the way to the molecular cell. They can affect the dendrites themselves, and then they will find the feedback coming from the Purkinje cells. All right, guys, so this is what happens at histological level. And this is the story between them, and you will find, of course, small cells that will interconnect, because we need to have connections between us. And some of these connection cells we have in the molecular layer is what we call the stellate cells and basket cells, and we need them because we need to make the Purkinje fibers connected all together. All right, so after talking about the gross anatomy and gross features of the cerebellum, we talked about the loops, we talked about what connects them to the brainstem, which is the